Welcome back. Good morning. My name is Alexander Krämer. I am a pharma and biotech equity analyst here at ABG. Today, I have the pleasure to host PharmNovo. We have the CEO, PharmNovo, with us, and the COO, Jesper Kjærulff, with us. The stage is yours, Pierre.
Thank you so much, Alexander. As said, my name is PharmNovo, and I'm the CEO of PharmNovo, a clinical stage drug development company. And as said, I'm presenting today together with Jesper, who is our COO, and also the project lead. PharmNovo's vision is to improve people's quality of life through innovative, sustainable, and effective pain relief medicines. And we're very proud today to be able to present the company and our lead candidate, PN6047, which is a phase 2 ready drug for neuropathic pain with first-in-class potential. In short, PharmNovo develops novel kind of drugs for neuropathic pain treatment. We have a team with extensive experience in drug development. Our lead candidate, PN6047, as I said, clearly has first-in-class potential, and we have patent protection until 2041. We recently completed and successfully completed the phase 1 study, and we are now targeting to go into phase 2A proof of concept studies by mid-2024. And for that, we are raising funds in a Series A led by Society in collaboration with ABG. Now, let's talk about the problem in the market. And the fact is that there is a huge unmet medical need for patients suffering from neuropathic pain. And neuropathic pain oftentimes ruins these people's lives, both socially, financially, and in many other ways. Neuropathic pain affects up to 10% of the adult population worldwide. And unfortunately, current treatments such as antiepileptics and antidepressants are only partially effective in some patients, and most of the time associated with quite severe side effects. When we talk about neuropathic pain, I want to just mention that the most challenging associated symptoms are allodynia or mechanical allodynia, which means that these patients suffer from the fact that light touch becomes extremely painful. The other classical symptom is hyperalgesia, and that means that something which is only slightly painful also becomes extremely painful. And these are common symptoms for the neuropathic pain patients. With that, I'm going to hand over to Jesper to take you through our solution and the development.
Thank you, Pierre. Yeah, my name is Jesper Kjærulff. I'm the COO at PharmNovo and also the project lead. Now, oops, that was the wrong direction. Yeah, as Pierre introduced, PN6047 is towards neuropathic pain, and it introduces, as we believe, a new area in treatment of neuropathic pain. PN6047 is a novel DORA, delta opioid receptor agonist, and it effectively, in animal models of pain, reduces neuropathic pain with allodynia and hyperalgesia. It is safe and tolerable in animal models, and as we'll go through later on, also in humans. And as Pierre said, we have first-in-class potential. It has the potential to become the first delta opioid receptor agonist in the marketplace. What is a delta opioid receptor agonist? There exist 3 different kinds or classes of opioid receptors. On the right-hand side, the green one is a kappa opioid receptor, and that is of no interest. It introduces a lot of unwanted side effects. In the middle, the red one is the classical mu opioid receptor, and that is what is normally targeted by classical opioids such as morphine and fentanyl, which is very effective in treating acute pain, but which is also associated with a lot of significant side effects such as abuse potential and also respiratory depression, which is the effect that actually kills people with overdose. On the left-hand side, there is a delta opioid receptor, and that is what we are targeting exclusively with our drug. Our drug is highly selective for the delta opioid receptor and has in reality no affinity from neither the mu or the kappa opioid receptor. And the delta opioid receptor is the receptor where we see the effect on neuropathic pain, but not on acute pain. Besides being very selective for the delta opioid receptor, we have built in an additional feature of our drug. In general, a lot of receptors can work through different signal pathways, and in terms of the delta opioid receptor, there are two main pathways. One is the G protein pathway, which is the one that we, with PN6047, target, and which leads to pain relief and antidepressant and anti-cough effect. The other pathway, which a lot of other drugs work on, is the arrestin pathway. And that is a pathway which is also associated with significant downsides, such as tolerance building, meaning lack of effect over time, and also seizures. But besides being selective for the delta receptor, our drug is also selective for the G protein biased or the G protein pathway, not for the arrestin pathway. The preclinical data we have in support of our drug is that the animal model of neuropathic pain, the SNL model, here you can see on the left-hand side, the gray bar, introduces pain when you make a surgery of the rat. Now, that pain is effectively relieved with increasing dosages of our drug, PN6047, and already at 1 milligram per kilogram, we see a significant effect. Besides that, of course, we have conducted the classical regulated tox activities, and we see that our drug is safe and tolerable in both rats and dogs, and now also in humans, which I'll come back to in more detail. A validating point of much importance is that the National Institute of Drug Abuse in the US has shown interest not only to the concept, but also in particular to our drug. Obviously, most people are aware of the opioid crisis in the US in particular, but also in Europe, and they are looking desperately for means to relieve that. First of all, looking, of course, for drugs which have no abuse potential, and here the NIDA has demonstrated with our drug that we have no abuse potential. On the left-hand side, you see a bar representing rats trained to self-administer drug, and in this case, self-administer a classical opioid because they feel the pleasance of that. Now, those rats, when treated or getting the offer to be treated with our drug, has no interest in that, demonstrating or validating that we have no abuse potential with our drug. These data are from NIDA, the National Institute of Drug Abuse in the US. On top of that, when people have been treated with classical opioids for a long time and withdraw from that treatment, they often get a lot of pain, neuropathic pain, or rather, more specifically, hyperalgesia. And that is often what prevents people from getting out of opioid treatment because they get these withdrawal symptoms, in particular pain. This graph, which is also, or this study, which also is produced by NIDA, demonstrates that when rats have been trained, or not trained, when they have been treated with classical opioids for a while and withdrawn, then they suffer from hyperalgesia. Now, there are two groups here. One is the control group. They have not been treated with opioid, but with saline, for example, and when they withdraw, they see no pain. Now, the red bar, or the red graph here, indicates that when rats has been trained or treated with opioids and are withdrawn, they get an increase in pain, illustrated with the first red dot here at the level of 4. Now, with increasing dosages of our drug, this pain is fully relieved. So again, we can treat one of the major symptoms of opioid withdrawal. Last but not least, also when withdrawing from opioids, you also get a lot of typically anxiety and depression. And again, these data has been generated by NIDA and demonstrating in this animal model that we can effectively treat depression with our drug. So altogether, no abuse potential, treatment of hyperalgesia, and also potential for treating anxiety and depression are data that NIDA considers very interesting. Now, let me take you through our data we have generated in phase 1. The scope of the study was, of course, to identify a dose and a dosing interval that is, first of all, safe and tolerable, but also has the potential, of course, to achieve an exposure which is predicted to be effective. Second, the scope was to validate our strong preclinical data in terms of safety and tolerability, and also that we are different from the classical opioids. We recently completed the phase 1 study, which was a relatively large study with 112 subjects. We included 3 different parts: a classical SAD, single ascending dose part, a classical MAD, multiple ascending dose part, and lastly a bioequivalence where we compared 2 different dosing formulations. This is a graph representing the exposure in the last MAD cohort, MAD 4, where we dosed 240 milligrams 3 times daily. This is the average concentration in the 6 subjects achieving the active treatment. And at steady state, at trough level, we reach an exposure level of 157 nanomolar. Why is this important? This is important because if we compare to the rat data in the rat SNN model, the pain model, we see maximum efficacy at a level of 50 nanomolar. So this illustrates that we have reached an exposure level which is predicted to be effective, and which, by the way, of course, also is safe and tolerable. So to conclude on phase 1, we have a safe and tolerable drug at doses predicted to be effective. We have seen actually AEs, adverse events, that possibly indicate target engagement. We have also demonstrated that our pharmacology is fundamentally different from the classical opioids. We do not see any of the side effects with classical opioids, such as respiratory depression, such as pruritus, such as constipation, which validates and proves that we do not touch the mu receptor and thus are completely different from the classical opioids. And last but not least, we also observe some characteristics that is suitable for a potential new drug in pain. So altogether, we are ready to move into phase 2. Now, how do our phase 2A plans look like? We have designed a classical randomized double-blind placebo-controlled crossover trial, which will investigate safety, tolerability, and efficacy of our drug. It will be in patients with peripheral neuropathic pain, but also specifically suffering from mechanical allodynia. This is the study design. As said, a crossover study where all subjects or patients will be treated both with our drug and with placebo, which altogether reduces the placebo response. Treatment duration is 20 days, with a washout period in between the 2 sequences of 9 days. We are targeting to recruit approximately 50 patients to achieve 40 completers. Obviously, we have discussed not only our drug and the pharmacology and the clinical strategy, but also the specific clinical study design of our phase 2 study with a range of clinical key opinion leaders, and they have all endorsed both our strategy in general and our study design and approach in particular. So the conclusion is now, from a development perspective, that we have a drug with a pharmacology that is fundamentally different from the classical opioids. We have some very strong phase 1 data. We have a very clear phase 2 plan. We have some very strong support from the NIDA, the US National Institute of Drug Abuse, for the opioid withdrawal symptom, and thereby also validating we do not have any abuse potential. And besides that, also we have recently applied for a grant from the European Investment for 2.5 million euros. And subject to that we get that grant, of course, that will also help us moving forward. So altogether, once again, we are ready to move into phase 2A. And with that, I will hand it back to Per.
Thank you, Jesper. So I will now lead you through the market opportunity, the IP situation, and some other company details. So we are looking at a chronic pain market, which is huge and growing strongly, expected to reach 140 billion dollars by 2033, which is a doubling from today. The specific neuropathic pain segment is about 10% of this, and already 2027 is expected to reach $8 billion. And if we look at our first niche indication for the phase 2A, that's about a quarter of the total neuropathic pain market. So it's a huge potential and a strongly growing market with an unmet medical need. The patent protection I mentioned in the beginning is strong, expected to have patent protection until 2041. And that's in all major markets. And these major markets shown here in green, they represent about 80% of the world market from a value perspective. I want to mention our team, which we have built up strongly over the last years. And today, we all together in the team have over 300 years of drug development experience from various pharma companies. The team members originate from AstraZeneca, from Lundbeck, Leo, Novo Nordisk. And I specifically want to point out that we have strengthened the team on the clinical development side with our CMO, Jarkko Kalliomäki, who is a pain physician and still active in pain clinics, and spent 12 years at AstraZeneca in pain drug development. And of course, Jesper, who has been speaking before me, has 20 years experience from leading projects from discovery to market at Lundbeck. We also have an experienced board, and I would like to mention Göran Lerenius, who worked 30 plus years at AstraZeneca and had a position as legal counsel pharmaceuticals. We also have Anthony Ford on the board from the US, and he has a background from Roche in the US and started Afferent Pharmaceuticals that was sold to Merck in 2016. So he has the full background from taking a startup all the way to exit. And we also have Per Anjansson, Karolinska Development, who is one of the main shareholders in the company, and Helen Pettersson, also one of the main shareholders from Almi Invest, on the board. Let me now just high level take you through the long-term development and financing plan. As we have described, we recently completed the phase 1 study very successfully, and we are now raising funds in a Series A, led by Society in collaboration with ABG, to finance the phase 2A proof of concept study that is already in preparations, but is targeting to actually start to bring patients into the study in the second half of next year. Following a successful proof of concept, we aim to seek partnership with pharma for further development and commercialization of PN6047, and that either through an out-licensing or through a trade sale. That would create a potential exit deal value, which is quite significant. We expect that our asset and company at the stage of proof of concept would be worth at least 5 billion Swedish. And that can be substantiated by numerous deals in our space, the pain space. And I will not go through all of this, but I would like to mention one of the recent ones, which is Eli Lilly's in-licensing of an asset in phase 1, actually. So a younger asset in the pain space from Comfort Therapeutics, and at a total deal value of $630 million. So to finalize and summarize, PN6047 and PharmNovo as a company is a great investment opportunity. We target a huge unmet medical need in a growing market. PN6047 has first-in-class potential. We have a strong support in the collaboration with NIDA and NIH in the USA, which we believe will help us strongly going forward. Our asset is phase 2 ready. And last but not least, again, we have a team with extensive experience in drug development that can make this happen. Thank you for your attention.
First of all, thank you very much, Per and Jesper, for this very nice presentation. Now we come to the Q&A part of today's session. And first, I would like to ask you if you could give us some more color on the design of the phase 2A study that you're planning now for next year. Which primary and secondary endpoints would you like to look at? And specifically, with a focus on which efficacy endpoints would you like to see?
Yeah, sure.
I will ask Jesper to do that.
Yes, I mean, it is a classical design for a phase 2A study. The primary endpoint will be safety and efficacy, which is a more descriptive endpoint. And the secondary endpoint will be a range of efficacy endpoints or efficacy measures. I think we have a total of 6 different ways of addressing efficacy. The reason we have chosen to do it that way is that at this stage, typically you don't know exactly which endpoint you would choose as the most optimal. What is the most optimal scale to be used addressing in particular our drug with our specific patient population? So there's a risk if you choose one of them upfront as the primary endpoint that we will fail on that and then end up with a formally failed study that could have at least significant signaling value. The advantage of doing it the way we do is that we can explore a range of different efficacy endpoints and then choose the one that appears to be the most sensitive and best for our specific drug and for our specific target population, and choose that endpoint then for the forthcoming studies as the primary endpoint. So it's a deliberate strategy of exploring now a range of efficacy endpoints without the risk of having a formally failed or negative study.
Thank you very much. A follow-up question maybe here. When you look historically, the FDA, which efficacy endpoints did the FDA require for historic phase 2b, phase 3 studies for approval of a new drug in that space, in that specific indication?
Yeah, in general, I mean, pain is an area where there are no objective measures of efficacy. It is subjective assessment by the patient. There's a range of different scales that can be used and which are accepted by the FDA. And the FDA also encourages actually development of new efficacy assessment or measures. The most important thing is that by the FDA that the method, whatever you choose or even develop, that it becomes validated, documented, that it actually addresses the issue of concern. But classically, it is a numerical rating scale, NRS, that on a scale from typically from 0 to 10, the patient values or addresses the pain intensity over a specific period of time or at a specific time point, coupled with a lot of additional patient-reported outcome measures that address typically the overall quality of life. All of this is generally accepted by FDA, the most important thing being that you choose a scale and a tool which is validated.
Okay, very interesting.
I think it might be worth to also mention that the selection of this patient category and also the study design here allows us also to explore something which we believe is very important, that is semi-quantitative testing. So there are today methods that have been developed over the last decades to actually semi-quantitatively be able to measure pain. So that's behind the strategy to go for neuropathic pain patients, which suits very well for those kind of complementary measurements.
Correct.
Okay, great. Then another question. You mentioned already in your presentation that there's a number of recent deals in the pain space. And I wanted to ask you, like, how does the competitive landscape look like in this space? Is there any other company developing a delta agonist like you are doing, or like beyond that kind of target, like other targets for this indication?
There are quite few delta agonists in development. We could mention that this was a large discovery and development area a long time ago, both by Pfizer and AstraZeneca. But they, as most people in the life science sector know, they actually basically discontinued the whole area of sometimes CNS and sometimes pain. So the area has been underinvested for quite some time, which means that there's a huge opportunity for new development. But talking about just delta agonists, it's only very few, and more correctly, it's one we follow, which is for anxiety and depression in Japan. But they are behind us in development, so we actually have the opportunity to reach market first as a delta agonist. Then there are numerous other kind of mode of actions in development for targeting chronic pain in general. And there are, of course, many subcategories in there. So we don't see that as a problem. It's a huge underinvested area and opportunities for many actually to develop new candidates.
Okay, great, thank you. And the last question, your phase 2A, you mentioned that you would like to recruit your first patients in the second half of next year. When do you expect to see results? Like what is the timeline, more or less?
Please.
We expect, we expect to have the headline results towards the end of 2025.
Okay, great. Thank you. With that, I would like to finish this session. I thank you very much, Jesper and Per, for this very nice presentation, and thank you very much for being here. And with that, we conclude the session. Thank you.
Thank you very much.